Yeah, good morning. Very good morning. Good morning, Akshya, Mithila. I hope today only very few members may come. So we'll have a quick doubt session, maybe 15-20 minutes. Okay. So in this, if you have doubts, you can ask me, and I will tell that what might be the possible model of questions you may expect. Within these lessons, not just cumulative tests, but even in final tests. Okay. During class itself, I'm explaining about the questions, the level of questions, or the type of questions. But now this is a time where we can more utilize on the what is a type of questions as well. Okay. Oh, am I audible? Yeah. Okay. So in last two classes, I already told yesterday itself by 6 p.m. if you can send any doubt topics, I would have discussed. But no one sent any doubt topics. No one asked anything. So today, whatever type of questions you may expect in these two lessons, we will try to have a small, brief understanding of that. So I've been telling that the statements that were given in what is a uh, for example, steps involved in plant breeding, right? So they may jumble up the sequences or the steps or the statements that are involved in poultry management, right? So those statements may come as what is a correct or incorrect statements. Okay. So likewise, we will see what are the other important aspects we have to touch in these lessons. Okay. So let's move with the first one, that is strategies for enhancement of food production. So in this, what might be the type of questions? What type of questions you may expect? Okay, let's deal with it. Just a moment. Just open the book. Have you gone through yesterday lessons? The lessons, biotechnology. Arshia, good morning. Hmm. Okay, so let's move quickly. For example, if you see this, you know, if you see the questions from strategies for enhancement of food materials. Okay, good morning, Abniti. Akshaya RP. Yeah, yes, you have gone through. Hope that models were really helpful so that you got a clear cut idea what you've been studying for over a year, right? So especially when I come to that biotechnology, I will tell why those questions are important. So let's see the strategies for enhancement of food production. What type of questions you may expect, right? I'm in our bio test, cumulative test. I am even giving the type of questions. It means the type of questions would be tricky, critical thinking aspects, memory based, right? Tricky questions that I already mentioning over there. That questions. So you should be paying extra attention to those tricky questions where they may look simple but you may go wrong. So For example, questions you may get like you know uh, for example, if you see this strategies for enhancement of food production, you may get questions from the different steps involved in plant breeding. What are the steps involved in plant breeding, right? And what might be the conventional plant breeding technique or animal breeding technique to the advanced breeding techniques, right? So conventional is nothing but very primitive or old. So likewise, this conventional breeding techniques, are right from conventional breeding techniques to even uh, the high-end modern technology like MOET. So you may be asked in that type of questions as well, right? And then for example, you may get definitions, right? Few definitions will be there, and the definitions you need to be identifying with the correct statements, right? And coming to so most probably what you will get is what are the varieties of different rice, wheat, right? And then what are the boxes, the mnemonics that I gave in the class, right? So those boxes are at most important, and you may get as an examples. So what might be the example of pest resistant in in this following example? What is a pest resistant variety? In that way, you may get questions. For example, silk rearing or honeybee. So their scientific names, right? Bombax mori, and you have got Apis indica when it comes to honeybee. So you have to even go through those examples of organisms which we are using in this breeding methods as well as food production. For example, management, that is beekeeping, etc. So those scientific names are utmost important so that you can cover it, right? And then how do we produce these MOETs, right? You may be shuffled with the procedures, etc., so that you have to put it in certain order. Like plan reading, also you have to keep it in order. If they shuffle the order, right? Uh, right. At the same time, you may be getting questions about the soma clones, micro propagation, and meristem cultures. Why we use meristem cultures in this way? These are very important aspects in these lessons. In this lesson, so one of which is that we will be mostly talking about the examples and their varieties. So that you need to more focus on this lesson the entire day, so that it will be more useful in the test, right? So if you have any doubts in this lesson, you please ask me right now, so that we can go for the next lesson. These are the hotspots in this lesson, Ra. That are those are nothing but examples, definitions, soma clones, micro propagation, right? Mutational breeding and outbreeding, outcrossing, right? Inbreeding depression, right? And MOET, what is a condition? Superovulation, how many ovules would form, ovum would form. So these are the aspects you need to focus on. So you have any doubts in this strategies for enhancement of food production? Biofortification, and what are the vitamins that we see in different crop products, right? Which institutions started this biofortification, which were given according to the NCRT? You need to more focus, right? Are you able to follow? So that's what about this lesson when it comes to the important areas or aspects of this lesson. Okay. But when compared to biotechnology, this lesson is, you have concepts as well as processes. For example, how do you produce MOT, or how do you do plant breeding, right? These are all processes, and they can jumble up, right? Okay, no doubts in this lesson, right? Are you sure? Shall we see the next lesson? Yeah. Okay. So these are the areas. I am only telling the areas. If you have doubts, you can ask me. That's what I'm seeking here. Okay. Yeah. Okay. Make use of time. Nothing more than that. That's it. So the next aspect is that the next lesson, what we're going to talk about is biotechnology, right? So what might be the questions you may expect in this lesson, right? You may be wondering that what sort of questions you may get, but I already gave three types of worksheets. In that, two types are pretty much almost 35 years, 32 years of previous year's questions. So you will get almost 32 years in your hand, and you can really get how they are actually asking. So that previous year's questions, the 45 or 50 questions are utmost important. You need to focus at any cost. Even if you by heart them, that's not a problem. When it comes to example type of questions, right? But when it comes to concept based, maybe the questions may not be repeated, but the concept may be asked. For example, active immunity, passive immunity. So that active and passive immunity over a 30 years, they will ask that questions from that concept itself, right? So 32 years or the 30 years worksheet, what I'm giving is at most important previous year's questions. That's why I gave even a action plan also. First online class, and then outline of the chapter, and then previous year's questions, and then daily questions, and then tests, right? So this is how you have to practice. I'm giving almost daily. You guys are doing in bio almost how many questions? 50, maybe 150 questions you are doing daily in two chapters, right? So that's what you are able to see different questions, and you are able to answer well in even cumulative tests. But the test marks are not that sufficient. You need to improve a lot. Okay, so let's move into the next aspect, next lesson, that is biotechnology, part one. Right, what might be the questions you may get? So I already gave the worksheets. You might have got a clear idea, but now let's again personally, like I will tell few important concepts that you need to focus on. Okay, so let's move to the biotechnology principles and processes. In this lesson also, again you will be getting critical thinking questions or tricky questions, diagrammatic questions you may expect from this lesson, guys, because this is rDNA technology, and you can't permit or you can't just leave those diagrams. In strategies for enhancement of food production, you may not have much diagrams at all, diagram based questions, but this lesson you need to focus on the NCERT diagrams. Like for example, how the production of transformant cells, right? There is a how do we prepare the rDNA? Once we prepare the recombinant plasmid, we introduce into the cell and we make it transform, and then we allow it to divide. So once it divides, then it actually the products we can expect from it. It means downstream processing, etc. So those diagrammatic questions are very important when it comes to this biotechnology. You can't really ignore any one of the line or any one of the diagram, especially the PCR procedure, right? How do we denature, how do we denature extinction and then annealing? How this process occurs, and what are the temperature? What are the enzymes? What are the substrates? For example, DNTPs, dinucleotide phosphates, right? This nucleotide phosphates we use as an reaction mixture. So these are all important aspects that you need to focus on. So especially the PCR is important, and then recombinant DNA technology steps, right from isolation of DNA to the transformation, 1 to 6 steps. They may jumble, and then they may give you as an in arrange in proper order, right? So diagrammatic questions are very, very important when it comes to this lesson, as simple as it. Right, and at the same time, what are exonucleases, endonucleases? What are their functions, right? Where exactly we use these endonucleases, exonucleases? I already told that endonuclease is the function of endonuclease is something to remove a piece of DNA. Within the DNA strand, there would be a desired gene that it can be taken out. That is endonuclease function. Endo, inside. Exonuclease means outside the cell. sorry, outside in the sense like at the ends of the DNA sequence. Likewise, these functions of enzymes, restriction endonucleases, and their corresponding functions. For example, BAM2, what are the specific sites it cuts, cleaves? EcoR1, SAL1, PUC1. So, PUE1, 2, all these enzyme restriction enzymes, how do they function, what are all their important sites of recognition, and where they cut the DNA. Those are the important aspects, and everyone would, or many of the people would go wrong in this question. So you have to make a tabular form and then you have to practice, right? What are the functions of gyrase, helicase, all these functions of enzymes we already talked in molecular basis of inheritance. So this biotechnology lesson is something which is you learn based upon the foundations of molecular basis of inheritance, right? Molecular basis of inheritance, right? You can't leave that sixth chapter, right? So likewise, you will be having which enzymes, or we tag polymerases got from Thermus aquaticus, like that. Which organism we use, because the biotechnology is nothing but usage of organism or the enzymes, microbial enzymes, right? So where we isolated these enzymes, different enzymes from different organisms, right? So these are the important aspects you need to cover, especially the processes, like what is gel electrophoresis, what is northern blotting, southern blotting, right? And autoradiography, and where you actually use this, what are all the processes, and you have to even identify processes, correct or incorrect statements. Okay, they may be asked as, this is a process of gel electrophoresis, and they give four options, and they may ask you to pick the incorrect statement, right? So diagrammatic basis questions, PCR, and you have got RDNA technology has got different examples where you have to more focus on the focus in this lesson, right? What is electrophoresis, why we use, right? So where exactly we use gel electrophoresis, right? So in which step of RDNA technology, this transformation of this, RDNA or transfer of this plasmid vector, right? In which stages we use. So these are the hotspots in the biotechnology and principles and processes. Because this lesson is more of a principles, you need to more focus on its principles, right? So, for example, within the RDNA technology, we use to amplify USPCR. So, what is amplification? So, for amplification, they may give few four statements. In that, they may ask you to pick the incorrect statement. Like that, the principles you need to more focus on, so that it will be more clear. And for example, what are the other for example, other different types of plasmids we use? What are the different type of cloning vectors we use? In what condition we use these cloning vectors are very much important. Okay, if the DNA sequence is short, which vector we use? The DNA sequence is very lengthy, which vectors we use? In molecular basis of inheritance, we already studied in human genome project, yaks, bats. So those are the examples you need to focus on, right? So this might be an approach that you need to have in this lesson, so that it will be more useful. So if you have any doubts in this lesson, please do ask me right now. Yesterday, whoever not attended the class, today is not the class. Today is only the doubt session. I'm not explaining anything today. I'm just giving the type of questions you may expect in future exam. Important model questions I'm discussing. That's it. Yesterday we talked about the lesson outlines, much more with models. And whoever not there in yesterday class, you can go through with the yesterday link. Okay, 9th and 14th, 9th and 11th lesson part 2. Okay, that video you can see and you can get benefited. Is there any doubts in this biotechnology lesson? Are you clear what we are doing? Okay, fine. You guys are already answered. Okay, fine. So this is the approach, guys. At any point of time, you can wake me in my sleep also. I will try to help you out. Clearing the doubts, etc. Okay, and and please do follow the class group because I'm posting every instruction in the class group very clearly. Okay, that is it for today. And if you have no doubts, we will end this session, and you please practice the two chapters, and you will be having 90 questions, right? By 5 p.m., we'll be posting the question paper, and by 8 o'clock, you have to finish the test and post the results as soon as possible. That's it for today, guys. And I hope this would be helpful for better it will give much more confidence to face the exams. So I hope you guys are clear. Okay, then see you then, guys. If you guys have no doubts, we can quit the session. Yeah. Okay, take care. Stay home. Stay safe. Prepare well. Ask me doubts at any point of time. You have my number. Okay. See you. Take care. Thank you.